To understand why postsynaptic mechanisms are so crucially important to the nervous system, let's briefly review how synapses are often classified. There are at least three sets of differences, each of which is largely explained by the properties of postsynaptic cells. First, there are fast and slow synapses. The former act in the order of milliseconds, while the latter often act hundreds, if not thousands of times slower. Second, there are strong and weak synapses. These are defined by the necessity of the postsynaptic neuron to integrate many weak incoming signals to reach threshold and be able to fire an action potential. But we'll talk more about this later. Finally, there are excitatory and inhibitory synapses. Excitation and inhibition are defined by the direction of the effect of the incoming signal on the postsynaptic membrane potential, and will be the main topic of the rest of this lesson.
Excitation and inhibition are largely defined by what they do to the postsynaptic membrane potential following synaptic transmission. Here we have a model neuron, drawn in yellow, which receives incoming synapses from a number of neurons. Consider the following experiment involving two presynaptic neurons, colored blue and red, synapsing onto the yellow postsynaptic neuron. We'll assign the blue neuron to be an excitatory neuron. The red neuron will be an inhibitory neuron. Let's look at the excitatory neuron first. As before, we're going to stimulate an action potential by placing a stimulator electrode in the blue neuron. And we're going to place a recording electrode in the dendrite of the yellow neuron to measure how the membrane potential is affected. If we were to initiate an action potential in the blue neuron, we can see that the membrane potential of the yellow neuron moves away from its resting potential of around -70 millivolts. It moves towards a more positive or depolarized value. The deflection of the membrane potential closer to the threshold makes it more likely for the postsynaptic neuron to fire. That's why this synapse is classified as excitatory, since it promotes the firing of the downstream neuron, allowing it to transmit a signal and continue the information flow. Call the resulting signal in the postsynaptic neuron an excitatory postsynaptic potential, or EPSP. It serves as a go signal for the postsynaptic neuron. Remember that ultimately it's the voltage at the axon hillock, where the axon joins to the cell body, that decides whether an action potential is fired or not. The effect of an EPSP at the axon hillock is different from what we record at the postsynaptic terminal. This is true for a number of reasons that we'll cover in a later part of this module. These factors include the shape of the dendritic arbor, distance between the synapse and the cell body, the number of other inhibitory and excitatory synapses, and the timing of the inputs, just to name a few. It's important to note that generally a single action potential in a presynaptic cell doesn't produce an EPSP large enough to reach threshold and trigger an action potential in the postsynaptic neuron. Instead, we need multiple action potentials, whether from the same presynaptic cell in rapid succession, or from multiple inputs simultaneously creating EPSPs in order to generate an action potential. Even though the final output is an all-or-nothing firing of the action potential, the integration of all the signals that the neuron receives and their modulation by different physical properties of the neuron contributes to the massive complexity of signal processing in the brain. To complement the excitatory postsynaptic potential, or EPSP, we also have the inhibitory postsynaptic potential, or IPSP. As you might have guessed from the name, IPSPs reduce the likelihood of the postsynaptic neuron firing an action potential. Let's consider the red-colored neuron, which we'll designate as an inhibitory neuron. Like the blue excitatory neuron, the red inhibitory neuron also makes a synaptic connection to the yellow postsynaptic cell. Again, let's place a stimulator electrode in the presynaptic terminal and a recording electrode in the postsynaptic side. Let's see what happens to the membrane potential of the postsynaptic neuron if we stimulate the red inhibitory neuron. What just happened? Nothing much, right? As you can see in the traces from our recording electrode, the membrane potential in the yellow postsynaptic neuron doesn't appear to change much in response to the stimulation of the red neuron. It stays at, or very near, the resting potential at -70 millivolts. This type of observed potential is called an inhibitory postsynaptic potential, or IPSP. It tends to prevent the postsynaptic neuron from firing an action potential, and as such can be seen as a stop signal for the postsynaptic cell. Even though it doesn't look like it's doing anything obvious from our recordings, the IPSP actually subtly changes the electrical properties of the membrane to work against EPSPs.